Good evening, ladies and gentlemen. For those of you who don't know me, I'm Trevor Macmillan. I have the great privilege of being the Vice-Chancellor here at Keele. And it's a great pleasure to see such a full room here this evening. In particular, I think a big welcome to those of you from outside the university, both colleagues of Rose and indeed, in particular, to various members of her family who've made quite a journey, I think, to get here today. Rose and I were just discussing that actually not all universities still do inaugural lectures, but we at Keele feel that they are still a very important part of academic life here. They give us a chance to show off the talent we have in the university, but they also give our talent their opportunity to actually give a slightly different form of lecture to the sort that they would normally give, in that they have to effectively address a very broad-ranging audience, those who are absolute specialists in their chosen field, but many people in the audience who probably don't have very much idea what they're walking into when they actually walk into the door of the lecture theatre. That makes it quite an intimidating period for any inaugural lecturer. These are quite difficult lectures to put together. But the very first good sign of that has to be the encouragement I get from an attendance like this. So again, I'm sure Rose will add her thanks, but again, on behalf of her, thank you once again for coming. Rose began her career at Bath University, student in applied biology. I should say I'm a biologist, so I actually recognise some of these words, unlike some of the inaugural lectures that I introduce. She had a Wellcome Trust PhD studentship at Cambridge under the supervision of Professor Steve Dunnett, who I understand is also in the audience, so welcome, Steve. And actually for that, she won the Brain Research Association Prize for the Best Biomedical Thesis in the UK in 1995. She was then successful in obtaining a Wellcome European Travelling Fellowship to work for 3 years at the University of Lund, where she was working in a group that was really pioneering cell transplant therapies. And in 1998, she moved to Harvard Medical School in Boston to spend 2 years working with Dr. Jeffrey Maclis, again on transplantation for cortical repair. She got a Wellcome Trust Career Development Fellowship to start her first research group at Cardiff University, and that's where her focus was refined to starting to work around stem cells and Parkinson's disease. She joined Keele in 2005 and has worked through from lecturer through to senior lecturer in 2009, reader in 2012, and this evening we're here to celebrate her promotion to professor. Her research group has been very well supported by a variety of different organisations, including Parkinsons UK and the EPSRC during that time. I'm not going to try and talk in any way about the research that work does, because she'll talk about that in a moment, but the fact that she has over 100 published journal articles, book chapters or conference abstracts, supervised 6 PhD students, been a member of the broader community, including Parkinson's UK Research Advisory Panel, and editor-in-chief of the international journal Neuro Report. I think importantly, even though we're here largely to celebrate Rose's research this evening, it's important also to mention the work that she's done in the context of the medical school. So along with her colleague, Dr. Coventry, she developed the first year of the current MBCHB program and actually co-led that course up until 2012. She then became the lead for the Doctor as Scholar and Scientist theme, and this summer she was appointed to the Director of Medical Science, responsible for a 25-strong team of academic staff who are delivering the medical science part of the curriculum. I think that is important to mention, because actually here at Keele we are very proud of the fact that our very strongest researchers can very much get involved in the teaching, and that in no small part is due to our great successes in things like the National Student Survey, which I cannot ever stand up on this stage and forget to mention. But nevertheless, a huge, therefore, contribution to Keele University. And I'm not going to, as I say, go through the various publications and things that I could mention. So all that remains now for me to do is to welcome Rose Fricker to the stage, our Professor of Neurobiology, and invite her to present her inaugural lecture, Stem Cells, Vitamins and Therapies for Parkinson's Disease. Rose.
I'll stand by this microphone while I sit and fiddle here. Thank you very much. Thank you very much for your kind words, Vice-Chancellor. And thank you so much to everybody for coming. I know some of you have come a very long way, and I'm absolutely thrilled to see such a huge audience. So what I want to do over the next 45 minutes or so, with a bit of microphone. Okay, that should work. Hopefully you can hear me now. Is really talk about my research career, pick out some of the highlights, and tell you how the field has moved and the great advances we've been making towards therapies for Parkinson's disease. So I'll touch on stem cells and vitamins and a few other things along the way. First of all, I thought I'd set the scene for you. So my research career started quite early on, and in fact, my mother was only talking about this afternoon. I was one of those children who asked very difficult questions a lot of the time. And so I always knew I wanted to be a scientist. But in fact, my interest in the brain, and particularly in Parkinson's, came when my lovely Granny Galpin, developed Parkinson's when I was a teenager. I then went off to Bath to study biology, and we did a lot of modules on the brain. I got very excited about those. And in the last summer, I got to work in the Babram Institute just outside of Cambridge for a chap called Dilip Singhi, or Singhi as we knew him. I was looking at things to do with the brain. One day he came in and said, "Oh, I've got this colleague of mine coming to visit, Dr. Steve Dunnett, his doctor as he was at the time. He's going to give a lunchtime lecture. Why don't you go along? I think you'd find it interesting." And in arrived Steve. Sorry, Steve, embarrassing photos of everybody today. And Steve gave this amazing lecture about what they were trying to do to transplant cells to try and treat Parkinson's and Huntington's, two brain disorders. I was absolutely blown away. And afterwards, the two of them came up to me and said, "We're going to take on a student this next year. We think you should apply." And after that, my course was really set. So why Parkinson's disease? Why is it so important to look at this and to look at therapies? Just to give you a little bit of background, here's James Parkinson. He was a doctor. He was also a geologist, paleontologist, and I think a little bit of an active parliamentarian as well. But he actually wrote an essay on the shaking palsy back in 1817. And he talked about the three main symptoms of Parkinson's you can see here: the shaking, the slowness of movement, and the rigidity or the stiffness that people show. But as many of my colleagues are here from Parkinson's UK will verify, there's also a lot of other symptoms that Parkinson's patients have, and they are often unique to the person. So they can lose their sense of smell, they can have sleep disturbances, and they have stooped posture, all kinds of things. And also some of them have mental health problems as well, anxiety and depression, and also some dementia. So it's a really debilitating disease. But that's not just the problem. There are 127,000 or so people in the UK now with Parkinson's, and of those, 95% are unpredictable. So you can't predict who's going to get the disease. It's not until they turn up with some of these symptoms and get diagnosed. And the other problem with that is that you have to have lost about half of the nerve cells involved before you show those symptoms. So the disease has already progressed quite a long way. And the biggest problem really is there is no cure. There are some drugs out there to treat the symptoms, but there really is no cure for Parkinson's. So this is why it was important to focus on this area. And just to show a little bit more background, this is the area in the brain that's affected. So there's some nerve cells that sit down here right in the deep in the midbrain, and they send very long processes up to another region of the brain sitting up here. And these nerve cells produce a chemical called dopamine. And these nerve cells are really like a stop-go sign. If you imagine you're out at a roadworks, and the man standing there flipping the stop-go sign. When these nerve cells are firing electricity, that's the go, the green bit of the sign. And when they switch off, that's the stop. And that controls our movement. So they really are the stop-go cells controlling our movement. And you can see it's a very tiny area of the brain, only a very few thousand nerve cells in there. But when they are lost, it's hugely debilitating to people with Parkinson's. So those are the ones that disappear, and those are the ones we want to replace. So when I joined the field in the early 90s, there was a basic way to replace cells, and what you did is you transplanted cells into this region here so they could locally release this chemical dopamine, and it would work if you used some nerve cells that could produce this chemical. And actually there had been a few clinical trials just starting in the late 80s that were showing some promise that this might work in patients. So that's the point I came into the field. And this was really the grandfather, I think Steve would agree, of transplantation. Professor Anders Björklund, who was at the University of Lund, where I was lucky enough to go after I finished my PhD. And he came up with a method, and his group and various others, to try and make these nerve cell transplants work. What they were doing in those days is they were taking embryo tissue, developing brain tissue, and they were taking out and dissecting out the tiny area that contains those nerve cells that make dopamine. You have to collect quite a few, and then literally you put them into a little container, you give them some enzyme to break up the tissue a bit, and then you really just mechanically dissociate them or break them up into single cells. And then you can transplant these back into the brain. So at the time, this was being done both tested in preclinical animal models and also in a few patients. Now, before I go any further, I would like to tell you that I will talk a little bit about animal models because they're some of the work that we've done in the past. And it's incredibly important to use them for this kind of research. The brain is such a complex place that if you're going to look at a technology such as transplants, there's no way we can model that in a culture dish. So we actually have to use the living brain to do that. And this work is always done very sensitively and with very much respect to the animals that actually contribute hugely to the science and taking it towards the clinic. So, I started my PhD with Steve. Oh, it was tough work. There was rowing, ballroom dancing. Very important that one was to me. And of course punting because we were in Cambridge. Then there were some fantastic conferences in far-flung places. Somebody can recognise the big house in the back here, probably. But apart from all that, I think we did quite a lot of science in those years, and I really enjoyed my time at my PhD. And I'm really going to sum up a few of the highlights of my PhD in just the next slide. And in fact, I wasn't working on Parkinson's at this time. I was working on a different brain disorder, Huntington's disease, but the principles are very much the same. What we worked out, and through work had gone before me and the work that I did in my PhD, that the best transplants had to make a connection with the host brain where they were sitting, and that connection had to be functional. The cells that were in the transplant had to talk to the brain cells, if you like. So this is just an example of what a transplant might look like in the brain. We stain to find the cells that we want, the nerve cell type that we want, and you can see this is the whole transplant. It's actually a bit patchy. So there are some cells that we want in the transplant, but there's also some cells that we don't want. And that's a pattern that I'll repeat a lot. You'll see that again and again. So this is our transplant. We've stained to find where the cells are. This is actually a marker for the host brain cells. And I hope you can see that it's also patchy, but those patches kind of line up with those patches. So what we knew from these transplants was that our host brain cells had gone in and connected to the right type of nerve cell. So that was a very exciting first finding. But what we did with these transplants is we wanted to look and see if the animals had some recovery. And what we did is we tested them on their movement, because this is a movement disorder. And we gave them a little task. They got to reach out with their paws and grasp for tiny little sugar pellets, a bit like pick and mix, if you like. And the animals love to do this. And what we do over 30 days is we give them 10 minutes to do this task, and then we just count how many pellets they collect. You can see down here, this is the animals that didn't get a transplant. And you can see this line is the animals who did, and they can collect far, far more, so they're very much better with their paws. So that told us that these good transplants were reflected in the animal's movement in recovering, if you like. And the final piece of the jigsaw puzzle that I did in my PhD is we had a collaboration with Hammersmith Hospital down in London, and they had a small animal PET scanner or a brain scanner. And we were able to take these animals down and have a look to see if their transplants were active. And that's exactly what's shown here. So this is a normal animal, and you can see these two white spots, the two hot spots of brain activity that we saw in the brain scan. If you take away the nerve cells on one side, you can see you lose it completely. And then when you give a transplant, some of that comes back. And you can see it's not as good as original, but there is some recovery. And this combined all together told us that actually When the graft and the host are talking together, then actually we get lots of good transplant success. So why can't we just do this with Parkinson's patients? Why can't we give them all cells? It would be a grand thing if we could do this. Well, there's some main problems, and this is what the field was facing at the time in the mid-90s. So some of the transplants might contain those unwanted cells, those ones that aren't the right type. And actually, in some patients, we've seen now that they can have side effects because of that. One of the biggest problems is cells don't survive very well. I showed you the technique where you broke up these cells and put them in a little suspension. In fact, that kills 95% of the cells almost. So actually, we get very poor survival. And that means for every transplant, you would have to take 6 or 8 human embryos to give to a patient to make a transplant that would work properly. And you can see that's practically very difficult, but ethically hugely challenging as well. So there's no way that the thousands of Parkinson's patients can get transplants. So at this point, my interest switched, because actually we needed another cell type. We knew the transplants might work in principle, but we needed a new cell type to go into the transplant. And this is where stem cells became exciting. This is around the early to mid-90s, and people were discovering stem cells all over, different parts of the body, different stages of development, lots of different types of stem cells. And they became very exciting to the field. They might be our answer. So why are stem cells so good? Well, they're very unspecialized cells, and their main job really is just to divide. So one stem cell will make 2, and these 2 will go on and they'll make 4 and 8 and so on. That's the main job of a stem cell. It's just to divide. It's very unspecialized. It doesn't do anything else. But stem cells can also have another property. They also give rise to specialized cells, and really any type of cell in the body. And they do that with a very neat trick. So if you take your stem cells, occasionally when it divides, it makes 2 different cells. So you can see here, this is still a stem cell, but now this one's a different one. And actually, just to give you an example of what happens in the brain, this becomes a brain cell, so it becomes slightly more specialized. But it can still divide a bit, but it's more specialized. The stem cell on the top will continue to go, make lots of itself. But the one down the bottom here will gradually go on to be more and more specialized. It will become a nerve cell, and eventually a mature nerve cell. And that's literally how the brain develops. So what you've got here at the end is a lot of nerve cells, but you might also have some stem cells if you're growing these in a culture dish. So this is what they look like. This is just one example. There are many types of stem cells, but this is just what I've picked out. These are from the very early embryo. So this is an embryo at 6 days old. And inside here is a mass of cells. You can take that out. They aren't blue. We stained them blue so you can see them. You take them out and you can put them on the dish. And they look like this. They're literally dividing. They're not doing anything else but dividing. And they're rapidly dividing, and they're unspecialized. So you can see that in a very short time, you can make lots of cells. So we could treat loads of patients with these cells. They're very easy to look after, generally. You just need to give them some food. Just like us, they need to eat. So they need sugars, and they need salts, and they need vitamins. They need all kinds of things. And often we give them something special to help them divide, special factors that stimulate them to divide a bit faster. But we like to keep them happy and healthy. They like to be warm. So like our cells in our bodies are at body temperature, our cells in the lab, we put in an incubator at 37 degrees. And we also have to keep them very sterile, because the conditions they like are the same conditions that the bugs like, the bacteria and fungi like as well. So we have to look after our cells quite a lot. But they're quite easy to grow. I'm looking at my students now, they're nodding. They're quite easy to grow, aren't they? And this is an example of how we can convert them into nerve cells. So when you get the conditions right, you can make some nerve cells. And this is a video that was taken by a PhD student of a colleague of mine back in the days when I was in Cardiff. And you can see here, there's some balls of stem cells. I don't know if you can see them here. These are unspecialized cells, but they found a surface to attach to. So if I play the video now, if you watch this ball here and see what happens, you can see these cells start to move around. They crawl out on the surface. But can you see they're also making lots of processes, getting very complicated. And they're actually converting into mature brain cells and nerve cells. So that's very exciting to us. This was in the early days, so this is around 2000 or so. We could see these cells doing this. Fantastic. We can take stem cells, we can make nerve cells. we can fix Parkinson's. Not quite that simple. So jumping forward a little bit, and I can see Jenny sitting in the audience, my first PhD student here at Keele when I arrived started to look at this to say, can we convert our stem cells efficiently into nerve cells? She, again, was using a slightly different model. She was trying to make a nerve cell that expressed this chemical called GABA. And so in the culture dish, she tried out lots of different methods to see what was good. And generally, she got some cells out at the end. I'll just explain a little bit what's going on in these pictures, because you'll see some other lovely coloured pictures. We can label our cells with very different fluorescent colours so we can find them again. So they're like fluorescent tags, if you like. So here is a blue tag where we can see all the cells. Here in the green tag, you can see the nerve cells. And then we can use a red tag for other markers. So we've used this one for a chemical called GABA. And the good thing is about using fluorescence is if you overlay them, they actually change colour. So wherever there's red and a green together, you get yellow. And wherever there's red and blue together, you get pink. So we can count our cells and we can work out who's done what and in what numbers. And Jenny found that in the culture dish, these cells did quite well. They made quite a lot of nerve cells, and some of those nerve cells actually had good electrical activity. It looked very exciting. And then she went to transplant some of them. So this is just a little bit of transplant data to show you. Here's a transplant on each side of the brain. The blue cells here, the transplant cells. Again, you can see some patches that are not all coloured in. We found a few nerve cells that look a bit like these ones. And we found that they had some of these markers that are the markers we were looking for. But I think what you can see here, and it's very important to see, this transplant here is very big. And if you can see, there's a little bit of blue up around here. That's because there's some dividing cells still in that transplant. So those cells have kept dividing, and they're actually forming a big mass in the transplant. So that's a big problem. We haven't converted all our cells into nerve cells, and that's very dangerous then to put into somebody's brain. So they could be quite successful, but not really. And so this is where the whole field got to about 10 years ago. We can only treat Parkinson's with stem cells if we can convert them, first of all, into nerve cells. And we really need to convert them all into nerve cells, so at large numbers to get enough for a patient. The other thing we need to do is actually make them into the right type of nerve cell. We've got lots of different nerve cells in our brain. If we don't get exactly the right type, it won't do the job properly for us. And this big problem as well, and this is what's really holding the field back and why there are no stem cell transplants in the clinic yet for Parkinson's, is that we need to control those cells when we put them into the brain. So we can't have any dividing cell in there that's going to go out of control and maybe form a tumour. So this is where the field was about 10 years ago. So then my research group decided we would take a little bit of a step backwards because we obviously didn't have a cell that was ready enough to test to go towards the clinic. We were obviously missing some things. So we went back to say, okay, well, obviously we're missing something here. We need to know more about what drives a stem cell into becoming a mature cell. So what drives a very unspecialized cell like this to get to something very complicated like this? And lots of other labs were looking at this at the same time, and they came up with, between us came up with two ideas. Either it's something internal in the cell, something like your genes or your DNA, that's, if you like, a program that the cell can follow to end up over here. There might be genes that can be switched on or switched off that will convert a cell from a stem cell into a nerve cell. A bit like if you program a computer to do a task for you. The alternative is, actually, it's the environment that's important. And particularly in a developing brain, it might be where those cells are sitting, those stem cells, that might tell them what type of nerve cell to become. And to us, this was more interesting. And that's really because of a couple of bits of work that I've done in the past. I'm just going to share one of them with you today. So this is some work I did when I was working out in Sweden with Professor Anders Björklund. And we had some stem cells, and we transplanted them into the brain, just of a normal adult rat. But we were a bit clever about this. We decided to put them into an area of the brain that has its own stem cells. So we as adults have two areas in our brains that have stem cells in them. And one area, hopefully, most of you are using right now, it's the area for learning and memory. So hopefully I'm teaching you something that you haven't heard before. And we've got some stem cells in there that will make nerve cells, because as you continue to learn and have memory, you need new nerve cells. this transplant actually went into the other area of the brain. And the exciting thing was that when we transplanted the cells into this area, and for the neuroscientists in the group, that's the anterior subventricular zone, those stem cells behaved just like the brain's own stem cells. So what they did is they migrated forward through this pathway here, and you can see here a couple of cells migrating, shown in this area. And then when they got to the front of the brain up here, they sort of moved out to the sides, and they became mature, and they put out processes. They looked like nerve cells, and they actually expressed some of the markers here. So our transplant cells are the green ones, and this green cell on the red cell is becoming yellow here. It shows it's our transplant cell that's changed into its fate, a bigger, mature nerve cell. So that was really exciting to us. So there has to be signals in that area that told our stem cells what to do. So that was one piece of evidence for us. So that's really what the lab started to focus on. And we had some fantastic funding from Parkinsons UK, two successive grants to look at this. And I had a very talented postdoc, Rowan Orme, who's been in the group for 7 years. And he basically said, are there signals in the developing brain that we haven't found yet? Are there things that we don't know that we should be looking for? And we basically went on a very big fishing expedition. We used a technique called proteomics to try and find those signals, because we thought the best way to do that was to try and find proteins in the brain that might be signals. So if I say to you, what is protein, some of you might say, oh, that's the lump of steak I have with my lunch. But in fact, our cells each are full of thousands of tiny, tiny proteins that all have very specific functions or roles. And so that's what we went looking for. We wanted to look for some proteins that actually we thought might be important in the signaling process. And so we took out the developing brain, the area here where the dopamine nerve cells develop. We took out that piece of tissue and we broke it up and we released all those little proteins. And then we went looking for them with some very fancy machinery. This is called a MALDI-TOF, and it allows you to separate out tiny amounts of proteins. And this is Rowan's famous analogy. If you were to take 120 kilo bags of sugar, tip them all in a pile, and throw in a grain of salt, then these machines could find that grain of salt. And not only could they find it, but they could tell you it's salt as well. So very, very fancy pieces of machinery, which we're fantastically lucky to have here at Keele. And we found quite a few things that were interesting. But the most things that were interesting to us were things that were involved in vitamins. So now I'm getting to my vitamin story. So a lot of the proteins we found were actually stuck on the outside of cells, on their membrane. And these things stick out, and they allow things to signal with them. So they act, if you like, they're literally like a satellite dish, like a receiver, and we call them receptors. And so what happens, if something in the neighborhood comes along that fits, it will bind. And that will change what happens in the cell. And we found a lot of our proteins were receptors for a lot of vitamins. So this is when we got very excited about what vitamins might be doing in our developing brains. So Rowan's work really looked at vitamin D3. I'm just going to talk about 2 vitamins that we looked at. And the active form of that vitamin is called calcitriol. So he basically took some dopamine nerve cells and he grew them in a dish. And some of them he added vitamin D, and some of them he didn't. So you can see here, these are the ones where he didn't. These are all the cells, and these are the dopamine nerve cells, the red blobs here. And these are the ones that had some vitamin D. Hopefully you can see there's more here. And if you count them, you give them different concentrations of vitamin D, you get an increase in the number of cells. But you can see after a point it drops off. And this we see a lot when we treat our cells with vitamins. That actually at some point they can become toxic to the cells, so they're not very good. So there definitely is a range where they work very well. But we could make more nerve cells, so that was very exciting to us. Now, Rowan did a lot of work to try and work out how those new nerve cells were getting there. And it turned out to be actually they were surviving better. So when we grow our cultures for 7 days, those cells actually survive a bit better when you've got vitamin D present. And to cut a long story short, he looked for the mechanism, and he found that vitamin D was actually causing the release of a specific molecule called GDNF. Now, I know some of you who belong to Parkinsons UK probably know about GDNF. It's a molecule that's now being used to treat some patients in a clinical trial down in Bristol. It's called glial-derived neurotrophic factor, and it actually protects dopamine nerve cells. And so at the moment, there's a few patients who are getting injections directly into the brain of this molecule to try and protect their nerve cells. The exciting thing for us was, if we added vitamin D, we could actually get our cells naturally to make this GDNF. So you can see the ones with have a lot more GDNF here than they do here, and that's just quantified over here. So the thing is, maybe we can give Parkinson's patients vitamin D, and that will actually naturally stimulate them to produce this GDNF molecule, which might be helpful to their cells. We've still got a lot of work to do to look at that, us and other groups, to see if this really happens. But we have some evidence to suggest in animal models that actually might happen. So we're very excited about vitamin D, and in a therapy in its own right, if you like. So while I was doing all this work on vitamin D, and we're getting very excited about this, I got a phone call from a chap in Birmingham, Professor Adrian Williams. He called me up one day and he said, "I've heard about what you're doing. Sounds really interesting. Have you thought about nicotinamide?" And I said, no, I hadn't thought about nicotinamide, but vitamin B3. He said, it's really interesting. I think it's really important in brain development. I think it might be one of your signals that you're looking for. And so I think 4 years later, after managing to collect enough money between myself, Adrian, and Professor Clive Hawkins at Keele, we managed to get enough together to get an ACORN studentship, and we got a student, Sheila Griffin, to come and work on this project. And she's just graduated last year. So what she did basically is to say, okay, what does vitamin B3 do to stem cells? She used a model where she was growing cells in a dish. So here's her stem cells sitting over here. And she had a method where over 14 days she could turn them into nerve cells. So it's a very basic method, but she got some nerve cells out at the end. And we had a cell line that actually, when the cells were dividing brain cells, they glowed green. They had an extra piece of DNA in them that meant they lit up when they were at this stage. So we could actually follow these cells in our cultures live. So basically, Sheila started off by adding vitamin B3 at various steps along the process to see what she got. And the major finding from her PhD was actually vitamin B3 is really good. It accelerates or it speeds up the process to get from a stem cell to a nerve cell. So just to give you a little bit of data for those who are the scientists in the audience, when she looked at day 14, the end point, she looked at her colonies to see what she'd got in there. And we've got some control cells here. In each case, the lower panel is the vitamin B3. And you can see here, there's less pink cells here, which means there's less stem cells at the end point. So she's lost a lot of her dividing stem cells. So if you remember back, that's very good for our safety. We're getting rid of the stem cells in our cultures. She also found that there are less of these dividing brain cells, these ones that glow green. So you can see a lot more here, but when you've had the vitamin, it's a lot less. So she's got a lot less dividing brain cells. And then the exciting result was she got a lot more nerve cells. So out at day 14, a lot of the cells have converted to nerve cells. And it seems like vitamin B3 really speeds up this process. It pushes them along the way, so more of them end up over here. But it only works when you add vitamin B3 right at the very beginning. And just a little slide for the Parkinson's people. The exciting thing was not only did she make more nerve cells in her dish, but she actually made more nerve cells that make the chemical dopamine. That's the one we want to treat Parkinson's. So you can see this is without the vitamin B, and this is with the vitamin B. You get over twice as many cells that make dopamine at the end. So that's a very exciting number to us. And I think those in the field would agree this is a very neat method. And in fact, vitamin B3 seems to be as good as all the complicated methods that are out there in other labs. So we're very, very excited about this. It might be a real tool to help us with our stem cell therapies. But it also might be very important in the developing brain as well. And that's something we want to further explore. And we've got a new PhD student, Emma, just started to have a look at some of these questions. So just to sum up the vitamin story, Sheila's work showed that vitamin B3 acts very early on to help this process from a stem cell to get to a nerve cell. But there are other vitamins that are important as well. So this is Rowan's work saying that actually vitamin D is at the end. It's very good. And work that I haven't been able to show you today suggests that vitamin A might help partway through the process, and vitamin C might help a bit later on as well. So you can see that clearly vitamins are really important in this process, which means they're probably very important in brain development, and that this might lead the field to go in all kinds of directions. So for the last bit of my research part of my talk, I just want to switch to something completely different. Now, if you remember right back in the beginning, I told you about animal models and the fact we need to do those because we need a living brain with that complex circuit to be able to test our transplants. Well, in fact, we're trying now to do that, grow that in a dish. It's not been done until now. And this has been in collaboration for probably more than 5 years now. I don't know where Paul's lurking, actually, in the audience, that I've had with Dr. Paul Roach at Keele University, a very talented engineer and chemist who's been able to build some very, very fancy devices. And we've had a couple of students working on this project, Mundu, who graduated last year, and Matt, who's now going into his 3rd year. And so they've been trying to literally rebuild a circuit of a brain in a dish. So this is the circuit. I've deliberately left this complicated. There will not be a test at the end, don't worry. But you can see this is what we would like to try and rebuild. This is the circuit that's involved in Parkinson's disease. And actually the dopamine nerve cells are the ones sitting in this box here. Each of these boxes is a different region of the brain, and you can see they're all connected by arrows. That shows you which nerve cells are talking to which one and which direction. But not only are there arrows, but there are also arrows of different colours. So in some cases, the nerve cells, where the green arrow is, when they talk to the one further down, that makes the cells switch on. Sometimes nerve cells switch off. That's the red arrows. So you can see this is a highly complicated circuit, and now hopefully you can imagine why that's almost impossible to build in a culture dish. But if we could do this, we would give a fantastic tool for us to use to screen therapies, both drug therapies and cell therapies. So that's what we thought it was worthwhile doing. So we started small. We decided we wouldn't go to the big thing. We'll just do 5. 5 will be easy enough. And I think, in fact, actually, we tried 2, and then we went to 5. So here's five regions that we wanted to put in the same dish, and we wanted to connect them up. So what we did is build this device, and Paul's team built this device. And you can see there are little wells here. You can put cells inside. But the different colours show that there are distinct areas where the cells can't mix. And I think this is here you can see in real life. So here's a template that Munya's made. This is his hand. This is about 10 centimetres across this whole disc. So you can see these are actually really quite small. And he makes a pattern with a technique called photolithography, and it's almost like a potato print, and then you can print out a stamp, and then you can make a mould. And the mould here is sitting, it's a polymer. It's a bit like very soft rubber, and it's clear. And you can see that these wells here, Munya's filled with coloured food dye. And you can see they're all distinct, so the colours aren't mixing. So that means if we put our cells in there, they shouldn't mix either. So that's very exciting to us. That's the thing is to design the device where the cells can sit. But the really neat part of the design is in these black areas here. What we've got here are tiny microchannels. These are 1/400th of a millimetre wide. Tiny, tiny channels, which we can make. Those channels are too small for a cell to go through, but they are big enough for a process to go through. So what we can do is we can have cells on this side growing their processes through to connect with the cell on the other side. And that's exactly what happens. So there's some pictures. Here's some cells sitting on one side. Here are the channels in the middle. And here are their processes growing out on the other side. And this is just another stain to show you the processes going into the channels here. So we got very excited that this looked like it could work. But we don't just want the cells to connect. We want them to talk to each other. And we also want to be able to measure that. And so we can measure changes in the dish. And the good thing about nerve cells is they make electricity. They're electric. So if we grow our cells on an electrode, we can measure that electricity and we can measure changes. So we've now combined our devices by putting something called a multi-electrode array underneath on the base that we can then grow the cells on top. So here you can see a tiny, tiny electrode with the cells sitting around it. Now actually we at the moment have a grid of 64 8x8 electrodes. So you can see here in this picture, there's a nerve cell sitting over here. This is population 1, and you can see some electrodes. Here are the microchannels, which also have some electrodes in. And here's the population 2 sitting over here. You can see the cells sitting close to them. And so we can measure from all those electrodes and tell if our cells are electrically active or not. And the reason we want to do that is we want to see if they're connecting and talking to each other. And the first results look very exciting. So this is electrical activity from all of those 64 electrodes. So you can see number 1 electrode over here, number 64 down there. And if I go on a bit, here's nerve cell population 1, that's all these electrodes up here. And here's nerve cell population 2 down here. And those are the channels in the middle. So what you can see here is lots of activity. So these are when the nerve cells are firing action potentials or creating electrical signals. But what you can also see is when the cells are firing up here, the cells down here are also making electricity as well. So we know that those cells have to be connected. So that's very exciting to us. So this is the stage we're at now, where we're trying to quantify and measure this so that we can make changes to these cells in these populations. Maybe we can make a model of Parkinson's disease by destroying some of the nerve cells, and then we can use it to test drug therapies or cell therapies. And the idea is that we might be able to give this to the pharmaceutical industry or academics to share around the world, and it would be a fantastic new tool to screen for therapies. So as the Vice-Chancellor said, part of my life here has also been at Keele Medical School. So in fact, half of my time since I've been at Keele has been at Keele Medical School. So I wanted to end by talking a little bit about the school, what I've done, and how far the school has come since it began, really. For those of you who haven't been there before, this is a beautiful med school building up on campus, and we have another one of these down at the hospital site as well. This is where we have our medical students in their first and second years. And our mission, really, since we had the medical school here, is to graduate excellent clinicians. And we've grown a bit over the years. So some of you probably remember the time before the medical school existed. These are our graduates in 2005, only about 14. You can see some staff have snuck in here. Some of us still, I think Margaret's still here. And this is our first head of school, Professor Susan Dilley, over here. And these are our graduates in 2016. We're up to about 130 or so medical students graduating each year now. So we've grown hugely in terms of numbers. And about a year after I arrived and started teaching in the medical school, the then head of school, Professor Richard Hayes, came to me and said, we're developing a new curriculum. And we think you might be one of the good people to try and lead this. And you'll have a friend to do this. That will be a new GP, Dr. Peter Coventry. Would you like to do this? And I thought for a moment, I don't know anything about curriculum development, but it sounds like a really nice challenge, so I'd like to do that. I'd like to give it a go. Thankfully, we had some very good steer and some sage advice and support from then Dr. Lindsay Bashford, who was our Director of Academic Undergraduate Studies. Because we really were quite taking a leap of faith here, because neither of us had that much experience in designing a program for medical students. But we set out, and we started in 2006, and the only thing we were really told by the head of school was, it has to be completely different to Manchester. So you can do whatever you like, just make it different. And that's partly because we'd been using the Manchester course, and we had to, for legal reasons, I think, it'd be different to Manchester. But we were to make it our own, to make it Keele's. So that gave us a lot of scope. And suddenly I was thrown in from being a person who sat in a darkened room looking at a tiny piece of the brain to suddenly leading a team of, we think, probably over 60 scientists and clinicians who came together to do this. We definitely didn't do all the work. It was a huge team event. We just sort of coordinated everything. We also had to stand up in front of the General Medical Council. They used to visit a lot and come and check us. They'd look at what we were doing. They would go and talk to our students to find out we weren't doing anything terrible. And Peter and I spent a lot of time defending ourselves. And I think that the biggest challenge was taking care of these 130 or so students, doctors in training that came in. And the most satisfying part of my job is really going from looking at a sea of faces of anxious students in their week one as they arrive, to seeing them at the end of year 5 when they're graduating as young doctors, confident and ready to go out and cure the world. It is a really fantastic job. It wasn't all tough. We have some fun as well. We have fun when we're teaching. So this is to show you a session of problem-based learning or small group learning that we do with our students. And here's one of our former PBL tutors, Dr. Lauren Brooks. But you can see the students enjoy this way of learning. So we get together. We teach the students some aspects of the course, but a lot of the course is really about the students learning for themselves and us giving them the tools to do that. And then we also have a bit of fun as well, occasionally. So this is a typical week 1 activity when the first years are coming in to get them to know each other and to know the staff. And this is one example of what we did at a Great Egg Challenge, I think. So often we would get them to launch eggs off a balcony without breaking them, or we get them to go through an assault course. So we have a lot of fun with them as well. So we've come a long way since that started in curriculum 2007. For me, I've come a long way. I've gone from being the co-lead of year 1 to a small time as being a science lead in Year 5, and then really overseeing the Doctor as a Scholar and Scientist theme lead, which I co-led with Sarah Hart until this year. So that's really looking after the science parts of the course. But our medical school has also come a huge way as well. So Keele is now in the top 10, and it's consistently in the top 10 for the National Student Survey. The students are very happy studying here. And last year, all of our graduates got foundation posts in the first round, which is exceptional for a medical school. We also know that our graduates are very well prepared for their posts. They tell us they feel prepared, so they fill in surveys to tell us that. But also the places they go tell us that they think our Keele graduates are well prepared for their jobs as junior doctors. So we're very pleased about that. But we're not resting on our laurels. We think it's time to now refresh ourselves. And so we're aiming for a curriculum refresh, review, and revise, so that we have a new look curriculum for 2018. So I thought I'd add this slide to really tell you where we all are now after those years. Professor Lindsay Bashford now is enjoying a well-earned retirement, doing a lot of hiking in the hills with lovely Barbara, his wife, and probably still earning a lot of money, collecting a lot of money for funds for charities. Dr. Peter Coventry, my Year 1 co-lead, is now the Director of Undergraduate Programmes, so he's really overseeing all of the undergraduate programmes in the school. And I'm privileged enough now to be the Director of Medical Science, so I am leading the team that are looking after all the medical science components of the curriculum. And Peter and I will be steering the ship, if you like, for Curriculum 2018. But it appears I've kind of lost the ship here somewhere. At least you can think I don't mind getting my feet wet. And the idea really is to look at having our curriculum to be research-led. We want our young doctors to go out there understanding the value of research, understanding the quality of research, and being able to use that in their practice. And for a small number of them, we would really like them to also to become the researchers of the future as well, to do the clinical research alongside their practice. So that's really all I have to say. I have to do a few thank yous. Thank you to all my collaborators and my team over the year, all those who've contributed to the science, you can see in various parts of the world, and to all of our funding agencies that have supported the work that I've talked about tonight, particularly Parkinsons UK, who've been a very strong funder of my research. And I want to end on some special thank yous. Now, to get to this point in your life as a professor, in my field, definitely, you don't do this alone. So my first set of thank yous are to my colleagues, my bosses, Andy, my current head of school, I don't know where Andy's lurking, and Andy Garner for being a fantastic dean. My mentors, Lindsay, Paul Eggleston, and Steve, and Anders, but especially the team. A lot of the work I've shown you tonight has been done by students and postdocs in my lab. So they're doing the hard work and pushing the science forward. It's fantastic. And my last set of thank yous are really from me personally. It's fantastic to have so many friends and neighbours in the audience tonight. And for those of you who know me, my path to get here hasn't been quite direct. There have been a few major challenges on the way. And I'm really grateful to all of you for the acts of kindness that you've shown me, particularly in the last few years. They're from very small acts to completely life-changing acts of kindness. And I will always be grateful for that. And my last thank you, and I'm going to embarrass them because they keep trying to embarrass me. They're all here tonight, so please. To my family. I don't think my three brothers, Robin, Neil, and Colin, actually know what an impact they had on my life and how grateful I am. They've always been there for me, particularly when times have been really tough. They will drop everything just to come and help me. So thank you all. And I want to leave on a last note. I'm really lucky in my job. I get to look at these beautiful cells every day. These are prize-winning photos by Rowan on this side and George, who's one of my current students. These are nerve cells and these are stem cells. And to me, science is amazing. And I hope some of the younger members of the audience tonight have been a bit inspired to think about a career in research. Thank you.